In this section, we will talk about the principle and application of Leica laser microdissection system. So, what is LMD laser microdissection? LMD is a contact and contamination-free method for isolating specific single cells or entire images of tissue from a wide variety of tissue samples. Therefore, you visualize your sample with a microscope and select under visual control specific area, such as single cells, cell cluster, for downstream analysis. And what is LMD principle? It is very easy. Leica laser microdissection is equipped with an additional laser axis to focus the laser beam through the objective to the LMD slice where the sample is placed. The sample is cut out by laser beam that is steered by prisms along the definition. It is collected by gravity without any additional force into a collection vessel of your choice direct positions below the slide. Why do we need LMD? In the beginning, in 1996, Emmerich developed a microscope-based microdissection system for accurate and efficient microdissecting cells from histological tissue sections. And why do we need LMD system? During the surgery, for example, in case of lung cancers, the starting material that is collected and used for further downstream analysis, like DNA, RNA, protein, in analyzation is always heterogeneous with every tumor slide. Always some healthy cells are collected and can mask the tumor cells in further analyzation. There is a microdissection is a technique to isolate pure and specific targets from microscopic heterogeneous samples to improve the downstream analyzation. Laser microdissection can be used for many applications, mainly in molecular biology, particularly nucleic acid research, neuroscience, cancer research, forensics, proteomics, plant research, for cutting cell cultures, and for single-cell isolation. Laser microdissection is even used in climate research, especially for examining the annual rings of trees. The fourth rule is always starting with a selection of a sample. The tissue samples can be embedded, sectioned, and stained according to conventional method of preparation. Partially sections, frozen sections, smear preparations, chromosome specimen, and cell cultures are all suitable for laser microdissection. Afterwards, the sample is stained for example for bright field illumination with chemotactic using staining, and also followed by the dissection of the region of interest with the LMD system. Then, the databases are prepared for further downstream analysis for DNA, RNA, protein, and analyzed, for example, with PCR. This video shows how the laser microdissection works. LMD system uses an upright microscope as base stand and takes advantage of the gravity to collect the dissected cells. This gravity collection is the gentlest and the contact and the contamination-free method. It can transfer densities of any sizes or shape and pull unlimited amount of specimen. If you need, you can fill the medium in collector. Then the dissectors collect direct into medium. This is a sample holding device. You can put the four slides. There are the collection device. Since the Leica LMD system collects densities simply by gravity, you can use standard device for their collection. Just use common molecular biology reaction device such as 0.2 or 0.5 ml chill caps, or 8-well or 12-well strip caps, which you already have in your lab. Collection device can be dried or filled with reaction buffer for culture medium for the LMD application. For the laser of LMD system, it is solid-state laser. The wavelength is 340 nanometer. Average pulse energy is 120 microjoule. Repetition rate is from 10 to 5,000 Hz. Laser actual control can be adjusted. It means you can adjust the cutting width. While LMD uses 349 nanometer laser, it is because that the 349 nanometer laser is not absorbed by protein and the DNA. It does not affect the downstream analysis. In the LMD system, laser is the most important part. There are the parameters that we can adjust in the LMD software. This is the light path inside the Leica LMD laser module. The main components to adjust the laser are the offset length, the actual diaphragm, and the scanner unit. The offset length used to adapt the laser focus position. The actual diaphragm used to influence the cutting width of the laser focus, and the scanner unit also to guide the laser beam. How does the laser actually work? The laser actually allows the direct control of the cutting width. If the laser actually is bigger, the width of the cutting line will be wider. If the laser actually is smaller, the width of the cutting line will be narrow. For the laser offset optics, when the visible light passes through a lens, the focus position is different from the UV light. You can see the focus position of UV light is upper than the visible light. So we can, by moving the offset lens forward and backwards, the laser focus can be moved up and down. This is the scanner unit. The laser is guided. using two prisms which can be rotated. Leica LMD moves the laser beam next the sample. Try writing your name on a piece of paper by moving the paper instead of the pen. You will find it is hard to do. That is why we move the laser next the sample for laser microdissection. Only Leica microsystems use high precision optics to steer the laser beam along the desired cut line on the tissue with prism. That means the Leica LMD cuts with higher possible precision and speed. In the LMD software, there are several cutting modes you can use. In Move plus Cut function, it is unique to the Leica LMD system and allows the direct cutting with the laser without drawing cutting lines in advance. You just keep the left-hand mouse key pressed down and draw the area of the specimen you want to cut out. The laser beam will immediately move along the line you described. In the LMD software, there is another cutting mode named laser screw. It cuts subsequently in Z direction. By lifting the stage between the laser cuts, even very thick and hard sample can be dissected step by step. In the laser screw basic mode window, in this window, the number of repeats means to repeat the cut of the defined shape according to the step size. If you want to cut an 80 micrometer thick section in 20 micrometer steps, you should enter 20 micrometer as the step size and 4 as the number of repeats. The first cut will be applied in the focus, where the shape was drawn. For the first repeat of the stage will be lifted 20 micrometer, and the next cut will be applied, and so on, resulting in 80 micrometer in total. In the expert mode, here, it provides the step count. So it means it allows you to enter number of the step count for dissection and to define the step size and the number of repeats for each step individually. For example, here the step count is 3. For the first step, step size is 10 micrometer and the repeats is 2. The stage lifts 10 micrometer and the laser cuts once. So the stage lift total 20 micrometers. For the second step, step size is 2 micrometer and the repeat is 50. The stage lifts 2 micrometer and the laser cuts once. Total 30 micrometer. For the third step size is 5. Repeats is 8. The stage lifts 5 micrometer and the laser cut once. Total 40 micrometer. This mode is very useful for dissect materials with different layers of different characteristics. These are the parameters that you can adjust in LMD software. Power is laser intensity. Aperture is used to control the width of the cutting line. Speed means the speed of laser beam movement. Basement balance is the parameter for adapting the laser setting to the different properties of the specimen. If you do not get a satisfactory result by changing the power and the speed values, you can try to increase the specimen balance parameter. Head current is used to adjust the final force of the laser. Pulse frequency, you can adjust pulse frequency within the range of 10 to 5,000 Hz. Offset is used to adjust the integrated offset length, which moves the laser focus into the image plane. The LMD's power, aperture, speed, head current, and the pulse frequency setting are tunable to the needs of your sample. For example, thick blind section require high power, while cutting radial mirror will require less power. Thin sections can typically use better cuts than the thick sections. The Leica LMD in this call center, the available magnification are 1.25x, 5x, 10x, 20x, 40x, and 63x. Please note that the 1.25x is just for specimen overview, not for cutting. All the objectives just designed for Leica LMD system, the transmission of laser light at 350 nanometer are higher than standard objective. This helps to cut out the target cells. So these videos show how to use 1.25x objective to create a fast overview image. Here is a create button. Select it, a window will open, and the system starts to create slides overview automatically. After finish the fast scan, the overview image will be visible in the window and also in the folder slides folder. It helps you to find the target cells or area that you want easily. How to select the objective you want to use? When you need to cut out large areas, you can use 5x, 10x, 20x objectives. For individual cells, 40x, 63x objectives are a good choice. For chromosomes, you might need the highest 150x objective. The 20x, 40x, and the 63x objectives are suitable for the ABC module. The Leica LMD system allows light cutting within bright field and fluorescence mode. These features in LMD BGR, Fluo Q, are all high UV transmission and especially for DAPI, FITC, and Texas red dye. In the LMD software, it provides an intelligent cutting mode named Auto Vision Control, which means it can define and recognize cells automatically. This is designed for collection of hundreds and thousands of cells. The ABC function can be used for bright field image and fluorescence images. Any number of objects with identical features such as color, shape, size can be marked for laser microdissection. In the auto shape setting windows, recognition method offers several ways to set the threshold for recognition, such as grayscale and color. When the color is selected, the individual RGB color range can be set. To remove artifacts of very small structures, there is a slide Remove Small Objects to the left of the preview image. If you need to avoid the recognition of structure at the boundary, which would be destroyed during cutting, check Remove Boundary Touching Objects. This video shows how the AVC module works. The cell stained with brown color was selected automatically, depending on the auto shape setting. These cells were cut out and collected in the collection vessel. There are many different types of consumers for laser microdissection system. The most common consumable are various membrane slides and frame slides. Petri dishes with a PN membrane are also used. For collect the dissects, you can use 0.2 or 0.5 ml PCR tubes or 8, 12 well strips. Frame slides consist of a metal membrane which can be covered with various membranes. The thickness of different membranes are not the same. For large area cutting, PN membrane and the PT membrane might be the best choice. Frame slides with PL membrane are mainly used for single cell and the chromosome dissection in combination with the 63X or 150 X fry objective. For the fluorescent samples, we put the 4 kinds of the slides: fluoro membrane, PN membrane, PT membrane, and glass slide under 40X objective with reflector tube to absorb the autofluorescence. Fluoro membrane offers less autofluorescence compared to PN membrane, but also PT membrane might be suitable for your fluorescent dye. The PBS membrane, not shown here, is also suitable for fluorescent sample. If you want to cut living cells, you need to culture the cell on Petri dish with PET membrane. Before cutting, remember to remove medium. Here the cells are just covered. Too much medium results the cell has to fall into the collector. For the laser setting, try to reduce speed and increase the laser power. Please notice that the highest magnification objective used for living cells cutting is 20X. For the application, Histopathology is a main application of the LMD system. For the application of histopathology section, the first step is to prepare the tissue that you want to absorb. The second step is that the tissue can be embedded in paraffin or flash frozen in liquid nitrogen, then embedded in OTC for following cryosection. Cut the tissue sections and mount it on membrane slide. Then do the fixation and the staining of tissue section, which are the same as classical preparations for histology. You visualize your sample with LMD microscope. The region of interest can be identified and marked automatically or easily manually. The ROI will be automatically selected and collected via gravity into consumables such as PCR tubes. You also can stain the tissue section with a fluorescent dye. Put the tissue section membrane slide on the LMD microscope, then create the RF, followed by laser microdissection. This is an example for histopathology application. They needed to isolate trophoblastic cells and endothelial cells for downstream analysis. This is another histopathology application. They want to isolate melanoma cells. Image A is before microdissection. Image B is after microdissection. This region had been cut out and put into the collection vessel. Image C is the membrane. It is the melanoma in collection vessel that we can visualize with LMD microscope. This histopathology application is associated with lung squamous cell carcinoma. They isolated normal branchial epithelium and the lung squamous cell carcinoma tissue for downstream analysis. They found that downregulation of selenium binding protein 1 is associated with poor prognosis in lung squamous cell carcinoma. Neuroscience is also a main application of the LMD system. There are at least several hundreds of different neurons in the brain. Leica laser microdissection system help you easily obtain sufficient cell sample quantities from your brain specimen under visual control. Simply identify your region of interest and directly cut the area with a guided laser beam for downstream analysis. Area can be distinct brain regions, single cells, or even subcellular structures. Here are some examples of detecting different cells in brain tissue using LMD. The left panel is new and stained with DLD in hippocampus. LMD system can easily detect these brain cells. The middle panel is the GFAP extra size stand with a Leica 488 in corpus caliber. The right panel is DDNF protein cells stand with size 3 in cerebral. LMD system can easily detect these different types of cells and cut out for downstream analysis. It is also useful for cutting fluorescence samples. This video shows cutting fluorescence sample using LMD system. Draw the area that you are interested and cut. This is another fluorescence sample. They damaged DNA using the laser beam and found that PAR and XRZ1 gathered at damaged site. LMD system is also suitable for live cell cutting. This is an example that they cut out the transfected fluorescent cells and collected with a petri dish for recultivation. In general, classical selection of infected cell followed dilution series take 2 months, but they just took 4 days. This video shows the live cell cutting. Remember to remove the median till the cells are just covered before cutting. Otherwise, cells are hard to fall to the collector. For the application of forensic science, a particular challenge faced by forensic scientists are impurities of all kinds. Using laser microdissection, it is now possible to separate the required cells from impurities. These cells can be quickly identified and isolated through laser microdissection. For the plant cells, the main challenge to overcome in sample preparation are the woody stem and the thick cell walls of plant tissue as compared with animal tissue. The laser used in the Leica LMD system is able to cut through even thick and hard plant materials. By obtaining information for different cellular levels, it is possible to gain a better understanding of plant reaction. This is the hard sample, tooth section. Leica LMD system still can cut out different structures of the tooth section for downstream analysis. The laser of LMD system can be used as a surgical knife to damage such as the organ, skin, neurite of animal. They damage the light sense organ in order to observe the change of its behavior. Thank you for your attention.